Okay, I think we can start now. If people are joining later, I will anyway do a quick introduction about Lucerna-Chem and our collaboration with Agrisera. So I will start with that and then hand over to Joanna. So thank you again to everyone for joining our webinar about mastering Western blotting optimization. So just a few words about Lucerna-Chem. So we are the Swiss distributor of Agrisera since 2021. Our company is already active since almost 50 years. And we are distributing almost 90 different brands here in Switzerland. So if you are joining from Switzerland, you will very likely know us already and already purchased some products from us. If you're abroad, then I'm pretty sure Joanna will be able to share with you the distributor list for your different countries that you're joining from. I will take care of all the people in Switzerland who would like to place orders or have any questions about products after the webinar. So overall, we are offering, as mentioned, from almost 90 different suppliers products, almost 4 million in numbers. And I'm very happy to be here today with Joanna from Agrisera, one of our esteemed suppliers. And the program will be split up in 3 parts. So today we'll be starting with part 1, focusing on the sample collection, protein extraction, gel electrophoresis, and protein transfer. Then we're also very happy to see that most of you registered also for part 2 and part 3 of the webinar series. So very happy to see that. And you will receive a separate invitation for the following meetings. Then it's already time to hand over the word to Joanna. So since we cannot be expert in all different fields and all different techniques as a distributor, we're happy to rely on our large network with the different suppliers. And Joanna truly is an expert in Western blotting with more than 20 years of experience in the field. And yeah, I'm very happy to hand over to Joanna to start our presentation. Joanna, thank you for being here.
Thank you very much, and welcome everyone for this workshop. I know that you're joining here from very different countries, not only from Switzerland, but all over the Europe and even from India. So warm welcome to everyone who wanted to join today, and I'm certain you will have a lot of questions. This presentation is built upon questions which I have received from researchers over the last 25 years. And I hope I will be able to address those questions in my presentation. And in case I have not been answering them, then you are welcome to wait until the question and answer session and paste them into the chat box. Of course, you can also interrupt me if you wish, but I find the method with asking questions in the end a little bit more efficient. And here we're starting with part 1, and just a reminder that there will be the other subsequent parts which Thomas was mentioning. So just a reminder of the dates, you have it in your calendar, and I hope you will like to join us. Shortly about our company, we are now entering into the dark period because we are located in northern Sweden, so our days are going to look more or less like that, while in the summer we experience 24-hour daylight. It's a really special location. And we are not at the polar circle yet. We have been established 40 years ago. So this year we are celebrating our 40th anniversary. We're very thrilled about it. And a lot of things happened during those 40 years. On the pictures, you will see the beautiful location of our antibody farm, and the second location in Umeå, where we are purifying the antibodies, doing quality control, and shipping them worldwide. Since 2020, Agrisera is part of Olink Proteomics. It means that we are not providing custom antibody services. We are developing our own catalog and producing antibodies for Olink platforms. And since last year, we are also a part of Thermo Fisher family. And you can rely on our products for detection of various proteins and compounds for plant and algae. We have also an excellent collection of epitope tag antibodies tested on plant samples and our high titer secondary antibodies. I mean, with one vial, you can do a whole thesis. So if you're interested, please check it up or ask us. We will provide more details. Agenda today, I will cover Western blot protocol until the blotting step in today's meeting, and will reveal to you things you may not be aware of, which can help you a lot in conducting successful Western blots. And in part 2, 6th of November, I will continue until the detection. There were some questions which were concerning part 2 of the workshop. So, how to visualize and measure specific protein in a complex mix. Of course, we are using a Western blot for it, but do you know how old this technique is? Please check here on this reference. It has been established September 1979 or published for the first time. So you're using a technique which has been around for quite a while. And why a name Western blot? We have Southern blot for DNA, we have Northern blot for RNA, we have even Eastern blot for post-translational modifications. And why Western blot? The lab which worked and published this technique was located on the West Bank, and it somehow fits to the rows of winds for the compass, and therefore the name. And we are dealing with, to obtain the material to work with, to obtain a membrane on which the detection of our specific protein is going to take place, we need to somehow get the protein to that membrane. And just a reminder of how a transfer stack is looking. Of course, this is an example of a wet transfer, and the goal is to obtain a copy, a mirrored copy of what we had on a gel. So if conditions of your transfer are not fulfilled for specific target protein you're working with, there the problem may begin if there is not enough of a protein on the membrane. And you will receive this presentation in PDF, so you will be able to go back, and also there are certain links which I included here, which I hope will be useful for you in the future. So please remember, you are after a mirrored copy of your gel on a membrane. And in Western blot, the antibody is doing a work. So here, just a brief reminder for you how the antibody is built and how it functions. So it is a Y-shaped molecule, which has a constant region, as you see the handle, so-called handle, and this allows purification of total immunoglobulin fraction using protein G or protein A. But the most interesting part in mammalian antibodies is the antigen binding domain. It's a variable region where the binding of the antibody to a target protein occurs. And the antibody binding is crucial for us to succeed in the Western blot. How is it performed? It's an antibody which is recognizing and binding to so-called epitope. And epitope can be from 2 to 15 amino acids long. And as you see here, there are two different types of epitopes antibodies are binding to. So we have linear epitopes, which are subsequent amino acid in linear structure of the protein. This is a linear epitope recognized by antibodies in Western blot done in the natured conditions, immunohistochemistry. But we have also discontinuous epitopes antibodies are binding to. These are quite exciting because, as you see here, two distant amino acids can be brought together and antibody may recognize such a discontinuous conformational epitope. And these epitopes antibodies are binding to in techniques which require antibody binding to a native protein like immunoprecipitation or CHIP or Western blot in native conditions. So please keep this in mind that an epitope can be anything from 2 to 15 amino acids long. And clonality is also an important term here because antibodies, as you know, can be polyclonal or monoclonal. And what does it mean? Poly means many. So an antibody, which can be produced in various species, not only mammals, but also birds like chickens or ducks, antibody is binding to pool of several epitopes. This is very important. So it means when an antibody is produced, then it will, in case of polyclonal antibody, result in a pool of antibodies which may bind to fragments of 2-15 amino acids long from a given target sequence which has been injected. So this is poly, and mono means one. So the antibody will recognize only one epitope. And they can be produced in different species, not only in mice. However, there is a misconception here that says that monoclonal antibodies are, for example, better than poly. One can never say that because it depends upon specific antibody. Monoclonal antibodies are much more expensive to produce, and they are mostly used in diagnostics. Of course, their detection may result in less background. However, if a monoclonal antibody is losing reactivity, then there is no signal whatsoever because that specific monoclonal antibody has been deactivated. In case of polyclonal antibodies, we can observe slow deterioration of a signal because, remember, it is a pool of different immunoglobulins which will be recognizing different parts of the protein. So epitope from 3 to 15 amino acids. And antibody choice must match species and application. This is where success of your Western blots is starting. I have been troubleshooting Western blots for the last 25 years, and I have seen every year several examples where wrong antibody is used on the wrong species and protein. What do I mean by that? It means that without checking if the sequence is present in the protein which is to be detected, we assume that the antibody is going to work based on overall protein sequence conservation between two species. This is not true because if the antibody is made to a short peptide, that peptide may be outside that identity range. So please don't waste your time, and before you purchase any antibody, make sure that it actually fits to your species and to the application. Because if the antibody is produced to a peptide buried inside the protein, then it is not going to work in immunoprecipitation. And such parameters can be predicted in advance before an antibody is going to be used. So your Western blot success starts actually not in a lab, it starts here. So save a precious time, and before you intend to use any antibody, make sure that it is actually matching your protein. You may say, but well, the sequence is not published on a supplier website. Yes, that's correct. It may not be published, but you can still send a question and provide sequence of a protein you aim to detect and ask, is this antibody suitable for my species, for that target protein? So how do we choose the right antibody? Here you have an example of a photosynthetic protein, PSPA, and of course, antibodies are not made to transit peptides, which in this case is cleaved off for the mature protein. And here in green, you can see 3 examples of from which peptides can be used to produce an antibody against this protein. So as you see, it's a location at N-terminal, middle of a protein, and very C-terminal. So the sequence, which is used by a supplier or a colleague who developed an antibody, that sequence needs to be found in a protein you are working with. And when is the antibody suitable for multi-species detection? There are such antibodies where one and the same peptide sequence is found in proteins from all these different species. These are so-called global antibodies, which Agrisera developed, which will save you also time and money, because one and the same antibody can be used on a very wide species taxonomic range. For our Rubisco antibody, it's hundreds of thousands of sequences which this antibody is recognizing. So there are such possibilities when one antibody can be used in a wide range of species, but please remember to check it first before you use the antibody. This question I have received for previous webinars: will knowing antigen sequence help to identify cross-reacting bands? No, not really, because the chosen antigen sequence is unique, as you saw on the previous slide, and we are doing a bioinformatic analysis to confirm that that sequence is only existing in a protein to which we are making an antibody. So only mass spec or sequencing can reveal which are the cross-reacting bands. So based upon the sequence an antibody was made to, it's not possible to actually identify these cross-reacting bands. You can learn more later in this blog post. And I also received questions from people who are ordering custom antibodies. Why premium screening before antibody production is started? Why is it so important? Because as you see here, we have serum from 10 different animals. And of course, many of them are already showing a band in the 50-55 kilodalton range, which is Rubisco protein. So before any antigen is injected, by Agrisera to produce antibodies to plant proteins, we are also always checking premium serum. This is the serum from animals which have not been immunized. And we are choosing the animals which have no bands in the range plus minus 30 kilodaltons from a target protein. Many companies are not offering this service, which may later result in some complications, because there may be already a band in pre-immune animal, not immunized, which is very close to your target protein. So as you see here, this animal number 4 was not immunized, and it already has 2 very distinct bands to some protein. So this is the information suitable for those who are going to order their own custom antibodies. It is very important in case of antibodies to plant proteins, because animals are fed with plant-based diet. So of course, development of antibodies to Rubisco is a very big problem. And now we are coming to the Western blot. The antibody visualizing target protein will bind, and we have 2 options. The antibody, primary antibody, is already labeled with an enzyme or fluorophore. Or option 2, which I assume most of you is using, we are using secondary antibodies, which, as you see here, are enhancing the signal because we have more molecules of secondary antibodies binding to the primary, and therefore it can help us to visualize proteins of low abundance. Western blot workflow has quite many steps, and we're starting with sample collection, protein extraction, electrophoresis, and protein transfer. And these steps I will cover today. And until we are happily recording the band, it may take around 24 and more hours. And I will show you which steps of this protocol can be optimized and in some cases even shortened, and still to obtain a very good results. Theory is very good, practice can be different. Here you can see examples of Western blots from 4 different laboratories. The protein is quite difficult to work with. It's a transcription factor. And the same antibody, primary antibody, has been sent to those 4 labs. All are using Arabidopsis thaliana. As you see, results can vary from no signal to actually detection of a target protein. They were using different protocols, and therefore the outcome can be very different. So before you go to the lab and start the Western blot, you need to ask yourself this question: how much do you know about the protein which you're aiming to detect? This is very crucial for successful binding of the antibody to the target protein. So your checklist is here. Biochemical properties, including isoelectric point, because that is very important. If your buffer pH is at pH of isoelectric point of your protein, it's not going to be soluble. Expression level, organ tissue localization, cellular localization. This needs to be checked before any Western blot is started. And I will show you and explain why. Amount of target protein. Over the years, we have learned that if protein is of abundance less than 10 parts per million, then cellular fractionation or precipitation is required for its visualization. And these are rather challenging proteins to detect. And you may ask me where to check such data. This is this beautiful database called PAX, Protein Abundance Database, which covers very many species, plants, also mammals and invertebrates. So I would like to encourage you, before you start your Western blot, to check protein abundance in that database. If your protein is on a low ppm range, then you will know already that the antibody is not going to detect it in a total cell extract. Here's another resource for people working with plant proteins, which is, but these are RNA expression data, which are also helpful. However, I am using PAX daily to check potential difficulties coming from the detection of the protein. Sample quality is of crucial importance. And in case of some proteins, there are some challenging steps to perform. For example, extraction has to be performed almost in the darkness because the protein may degrade. Or for some proteins, there may be a need to perform extremely quick extraction to avoid the risk of degradation. And following this link, you will find some protein extraction protocols based on published data. So keep in mind that sample quality is crucial, especially of proteins of low abundance, and definitely do not store your samples in a freezer for several months because there is a high risk of protein degradation, which can later contribute to lack of band on your blot. So we have a high quality requirement for protein samples, and they can be obtained by different ways. Some laboratories are using bead beater, which allows to process multiple samples in a very short time. And we have more time-consuming extraction methods like mortar and pestle and sonication. In case of sonication, there is a high risk of heat generation, and mortar and pestle is a method which is rather laborious and tiring, especially when multiple samples have to be processed. So you need to investigate this before you start and optimize your extraction method depending upon the protein you're working with. We summarized information regarding the protein extraction in this poster, which is available on our website for download. And we also have hard copies, which are available, and you can order them on our website. And we hope this poster will be helpful for you to choose and optimize the most optimal method for your protein of interest. When we work with proteins of low abundance, there is no way that we will obtain a signal from a target in a total cell extract, as you see in this graph. Many antibodies have been discarded because of that. Protein fractionation is quite laborious, and however, it is worth the effort. Here you can see the example of the protein which the target has been concentrated with the subsequent fractionation steps. So keep this in mind. If your protein is of less than 10 ppm, you will have to fractionate, and this will allow you to concentrate the target and visualize it later. Protein extraction methods, of course, involve a choice of a specific buffer with a most optimal composition. This is a result obtained by one of the test labs, and as you see, that changing the buffer, the target protein band was visualized. So this has to be thought through before. Very many labs are using tree-spaced buffers, but HIPPIs seems to be more optimal in case of some proteins. And here you see so-called garden blots. We have samples from barley, Arabidopsis, and rice. And the peptide, which was used to elicit this antibody, is perfectly conserved in all three species. However, reaction was only visible for Arabidopsis, unless the urea, 6 molar urea, was included in extraction buffer, and it allowed to keep the target protein unfolded and therefore allowing the antibody to bind. So you need to pay attention to buffer composition, and if you work with proteins of low expression, you need to have specific isolate proteins from specific organs, include protease inhibitors, but in many cases also proteasome inhibitors, because some proteins may be targets of proteasomal degradation, and this is worth to check before you start extraction. You need to, of course, optimize your extraction method for a good yield. So the amount of buffer to tissue which is processed needs to be optimized. And I heard an expression from my colleagues that the ratio between extraction buffer and the tissue should allow to obtain a liquid, pancake-like liquid when extracting the proteins. And depends in which country you are making the pancakes. It can be more dense or less. However, if you're working with tissues which have high water content, of course, the proportion of the buffer to the tissue is going to be different than if you are extracting proteins from seeds. And you need to sometimes use specific conditions. And this has to be investigated before you go to the lab. There are numerous databases which will help you to check protein abundance, as I mentioned, packs. Then we have databases which will help you to reveal if protein is a target, may be a target of proteasomal degradation. So please check this before you start Western blots, because it will pay off in the long run. You will proceed to successful detection much faster. And is the antibody always to blame? As you may suspect, it's not always the case. As you see on this slide, sometimes it is actually an extraction buffer, which has to be adjusted to allow successful detection. And we need to work with good laboratory practice, which means we work clean, cold, and precise. These are crucial steps for your successful blots later. And I know that some labs may not perform their Western blots in the cold, but this is actually quite important. I will talk about it later. So please remember about the GLP. There are different precast gels which can be used. In our lab, we are using precast gels because it allows us for higher reproducibility. And which gel to choose, one can be a little bit lost, but as you can see here, they can differ in percentage and composition, and therefore offer you different molecular weight range, different pH stability, and shelf life. So depending upon if you are working with quantification, for example, the precast gels will offer a little bit higher reproducibility compared to self-cast gels. And as you see in this example, there can be a slight difference in intensity of visualized protein. Then we have gel electrophoresis. Sometimes in case of self-cast gels, there can be issues connected to polymerization process, for example, and the shape of the bands can be influenced. And what do we load per well? This is actually a very crucial step in Western blot procedure. Here you can see a gel prepared for electrophoresis in our lab. We are using Novex gel system for the last 25 years, which works very well in our hands and provides a high resolution. Depending upon the buffers which we use, we can change molecular weight range at which proteins are migrating. So we can use the same gel, but by adjusting the buffers, we can obtain a different resolution. So what do you load per well? Protein, chlorophyll, or volume? I do hope that you are loading micrograms of protein or micrograms of chlorophyll for photosynthetic organisms. Not recommended is the load per volume, because even though the source material may be the same for various samples, however, extraction may not be having the same efficiency. So loading per volume is not really recommended to obtain results which are reliable. And I often hear that, but how to measure protein concentration in the presence of components of extraction buffer. And there are actually regions which allow that, which are compatible with detergents, buffers, salts, metal ions, chelators, and so on. So I strongly recommend you to load based on the protein to be able to compare and analyze your results later. Absolutely not per volume. And one of the questions I received was if we are to load by nanograms or micrograms. And it depends what sample we are talking about, because if we're talking about recombinant protein, then we will load by nanograms. And if we talk about a total cell extract, we will be dealing with micrograms. And here you can see how we can titrate with a varied protein load per well and antibody dilution, how we can find the optimal load per well combined with primary antibody dilution. But this is, of course, a situation when the antibody is characterized and we found out conditions of protein extraction and successful Western blot. So these experiments you can do later, and you can simply titrate away the background. The other question I have received was about a difference between so-called one-dimensional and two-dimensional gels, and it depends what questions you are asking. In one dimension, you need to keep in mind that we have hundreds of proteins in each band. Proteins are migrating based on their molecular weight, so the heavier will get stuck at the top and the smaller ones will continue to the bottom. And in 2G gel electrophoresis, each such band is then later resolved into several spots. So we are actually getting another resolution, and use of two-dimensional electrophoresis depends upon your biological questions. There are Western blot results, of course, for these types of gels, but they are much less common. They can be hundreds of proteins analyzed at the same time with two-dimensional gel electrophoresis. Sample denaturation is another step in your Western blot protocol. And I wonder in what temperature you're denaturating your samples. Is it most common, what I see is 100 degrees, 5 minutes. We are using 70 degrees for 5 minutes. And for some proteins, samples should not be treated with 100 degrees because this can lead to protein aggregation, and as a consequence, your target protein will aggregate and get stuck at the top of a gel. So it will never enter the gel, and therefore there will be no band on your Western blot. So please take this into consideration. What is the way of denaturation of your sample? Protein transfer conditions are here presented for wet transfer. So we have so-called sandwich, and there are also different parameters which have to be considered here. We are always doing a transfer in the cold, in the ice bucket, and with cold buffers. Applied current makes protein to migrate from the gel, where they have been separated based on molecular weight, to the membrane. And always in the cold. So protein transfer conditions and size of a protein are actually connected. So here you can see conditions for transfer of proteins of molecular weight higher than 20 kilodaltons. And as you will see here, recommended membrane size is 0.45 micrometer. If possible, I would advise to have a membrane of both pore size in the lab and change it depending upon which protein we are dealing with. A bigger pore size will make it easier for the proteins during the transfer. Also, higher molecular weight proteins may require more SDS, and supposedly wet tank transfer may be more optimal here. On the contrary, low molecular weight proteins will require smaller pore size of the membrane and less SDS. Of course, the outcome of a transfer will also depend upon which protein you're working with. And when I was asked which transfer is better, it is not possible to say that one transfer is better, wet transfer is better than semi-dry, because it really depends upon your target protein and the protocol which was developed. And which membrane you are using, PVDF or nitrocellulose. There are some differences between them, and I will show you which they are. So there's a different mechanism of binding for PVDF compared to nitrocellulose. Both membranes can be air-dried, and PVDF has better protein retention. It's more robust membrane, and I can see that majority of laboratories is using PVDF. But for some protein antibody pairs, better results can be obtained with actually nitrocellulose. So I would recommend to have both membranes in the lab if possible, or if something does not work for your protein antibody pair, maybe try to get the other alternative membrane to try if the result of protein transfer will be better. Both membranes are available in those different pore sizes. Membrane activation. First choice is, of course, methanol. However, I have been asked if ethanol can also be applied because it's less toxic than methanol. And in our lab, we have switched to ethanol and have not observed big impact on the quality. However, if you want to make this change, you have to test it for your protein antibody pair. Protein transfer type, we have two types, wet and semi-dry, and they differ. So with semi-dry transfer, we use less buffer. There is a direct electrode contact and it goes much faster. However, semi-dry transfer may require a little bit more skills to set it up and make it working. And for beginners, wet transfer is recommended. And also, when you are assembling your sandwich for the wet transfer, I would suggest to do it directly in the cassette and absolutely do not roll out the air bubbles as showed on this picture, because this can actually contribute to the membrane rubbing the gel and moving. So if you assemble your sandwich directly in the cassette, as shown on this picture, then you will be able to control air bubbles also here in a better way. And if the air bubble, which we see, is located outside our target protein range, then in some cases we will not try to desperately remove it. We made a short video for you how to remove the air bubbles, which you can watch later. But of course, the sandwich has to be tightly secured in the cassette, and you should never rub and move it around, because that can contribute to quality of the transfer later. After protein transfer is done, we are checking the quality of the transfer, and we can use both stains Ponceau or Comasi R250 to check how the membrane is looking. So, of course, we will not continue if the transfer looks like on the picture here. We want to have a consistent and nice band pattern. And both Ponceau and Comasi are both reversible stains. This quick check we do when we are working out a protocol for a new protein extraction, for example. This more important will be to check actually a membrane, a gel later after the transfer. Because if you have any bands left in a gel after you open the cassette and disassemble the sandwich, it means that your transfer is not good enough. And as you see here, we used a double sandwich here. So we had 2 gels and 2 membranes in the cassette, and we observed protein bands on a gel from a second membrane. This is a very quick to stain the gel, so I recommend that you do it to check if your transfer was complete. In some cases, that for proteins of low molecular weight, a transfer time needs to be adjusted, for example, shortened. I troubleshooted many antibodies for low molecular weight proteins which did not give any signal on the membrane due to that the protein was blasted through the membrane. So keep in mind that for low molecular weight proteins, you need to have shorter transfer compared to high molecular weight proteins, which may require longer transfer. So this is a parameter which you can work out preparing your protocol. And we are coming to antibody format. What you see here is a lyophilized antibody, which is going to be shipped from Agrisera, but we have different formats. So be aware of differences between those formats, because we can purchase antibodies in serum or purified to total immunoglobulin fraction or purified on a specific column to obtain antibodies specific to the target protein. So often we receive the questions, how much of the specific antibody is present in serum? And one cannot answer this question until antibody is purified. There is a difference between those formats. So if you see that the antibody which you aim to purchase is provided like 0.5 milligram per vial, please check if it is affinity purified or it is most likely a total immunoglobulin. Antigen affinity purified antibodies will contain the most of the purified antibody in the tube. Usually it is between 0.5 to 5% of total immunoglobulin pool specific, which will be for specific target protein. It depends between different antibodies, how much of a specific antibody will be produced by immunological system of the animal. And primary antibody can be subjected to antibody purification. This is a setup which we are using in the lab, where we are working with antibody purifications on the specific antigen columns. So the column with antigen is prepared. And just to remind you how the process is conducted, so we have a specific resin on which antigen is immobilized. So it can be a peptide or a protein which is immobilized on the resin. We are passing the column, passing through the column the serum, which contains specific antibody, which is going to bind to the antigen, and then elution is usually done with low pH. This process can also be done as a bench purification in macro or micro scale. It depends how much serum is available. Elution results in antigen-specific antibodies, and if you ever produce the antibodies, please keep in mind that never whole serum should be purified at once, because it has happened that some antibodies may actually lose activity after purification, and they are most stable in serum format, where there are other proteins which will stabilize them. How long can the same aliquot of antibodies in blocking buffer kept in -20 degrees be used? Some labs are doing reuse of primary antibodies. They are expensive reagents, and it depends actually strictly upon the primary antibody if it can be reused. Some antibodies can be only reused once, and some can be reused quite many times, as you see in this example. But you need to test it, and before the antibody is fully characterized, I would not recommend you to reuse it. This depends strictly upon the antibodies. So please keep that in mind, and if you are disappointed that in a second blot with the same antibody solution there's no signal, it means that your antibody absolutely cannot be reused. Primary antibody may sometimes stop working. Maybe you have experienced this, and there are different reasons for it, that the antibody can stop working. And here's a quick checklist. Sometimes storage temperature was not appropriate. The antibody may lack stabilizers. And in such case, if you purchase the antibody, you should always check product information sheet. What are recommendations of the storage? There was a lab which always stored the antibodies in a certain way and didn't check product information sheet. And addition of glycerol to serum is also not necessary. So if you use catalog antibodies, please always refer to product information sheet to check for appropriate storage conditions regarding temperature and additives. And sometimes antibodies may lose activity after a couple of years. So we had such cases. Some antibodies may be active for 20, 40 years, while others will lose activity after 1 or 5 years. And these are biological molecules, and this process cannot be predicted in advance. When it happens, unfortunately, such an antibody may be in a need of being discarded. So especially monoclonal antibodies, when they lose activity, then there's not much to do. In case of polyclonal antibodies, as I mentioned, you can observe a small, slow deterioration of the signal, which can be a sign that the antibody is deteriorating. And there is a second educational poster, which you can also receive or download from our website, which is focused on Western blot. And this poster is my attempt to summarize those webinars. And I hope it will be useful for you in the future. You can easily order it from our website. It's free of charge, or download it for your own use in the future. And I hope it's going to be helpful. And here is a surprise number 1. We have over 153 antibodies, which we offer in return for results. There is no limit on how many antibodies we provide. And we are also including secondary antibody together with the primary. So check the list and tell me if it fits your experiments, if there are antibodies which you would like to use. And you are also having a chance today to win a free antibody of your choice. And here is a QR code to a short quiz from which is covering the material I was going through today. So you can do it later after the webinar. and we will choose the winner tomorrow. I will paste the QR code in the chat and the link to, or you can make a picture right now with your phone. And for contact in Switzerland, we encourage you to mail or ring Lucerna. And for contact with questions from other countries, you're welcome to contact Agrisera. And with this picture of the autumn in northern Sweden, I thank you for your attention and I welcome your questions. So, any questions? You can type them also in the chat. And here comes QR code for the quiz. The tips to detect phosphorylated proteins. Here is the question. Yeah, phosphorylated proteins are a challenge to detect. And laboratories which are working with detection of phosphorylated proteins are often using chemiluminescent detection, for example, as more sensitive method. Nowadays, most of the labs have CCD cameras. Some laboratories we collaborated with were using film as more sensitive to record phosphorylation. Also, extraction protocol is crucial, and it has to be done in a very prompt way because we are after a very specific modifications. And of course, the quality of the antibody is of crucial importance. It is very difficult to make antibodies to phosphorylated proteins. It is a very challenging job, and the best company on the market, which is providing antibodies to phosphor modifications, is Cell Signaling. Any other questions? I'll be happy to answer. So please, there are no bad questions. I'm open to any questions you may have. And when it comes to phosphorylated proteins, I would actually visit a lab which is doing successful detection, because there are many tricks which can be revealed when actually when we participate hands-on and are witnessing the protocol with our own eyes. So if you have such a possibility, I would highly recommend it. You are also welcome to send the questions for the part 2 and 3. So if there was something which was not answered, I can take it later. You showed a slide regarding efficient protein load. I didn't really understand the difference of just adding the same amount of protein and the right side of the slide. Okay. Yeah. We had once a problem to detect a protein. We used 4 different antibodies and they showed us 4 different band size, though they were supposed to detect the same protein. How can this be possible? Yeah, it is possible. Let me address the first question about the efficient protein load. I will go back to the slide. It was about the titration of the protein load. I think you meant that slide. So in the left part, the protein load per well was the same. The antibody dilution, primary antibody dilution, was different. So that way, the background signal was also getting less intensive. In the right part, we have 2 different protein loads combined with different antibody dilutions. So this work we can do once the antibody is characterized and we know its performance, and when it is not detecting several different bands. And to address the second question about 4 different antibodies detecting different bands, I would start from analyzing to which parts of the protein these antibodies were made. Were they covering C and terminal? Were they from the inside of the protein? The question would be also, is it possible that this protein exists as several isoforms? So such situation is actually possible. And if you want, I can give it a bit more thought if you give me a little bit more information by email. So you can send me the more data by email, because I assume that these antibodies were probably done to different parts of the protein. And this could lead to a different band pattern later when conducting a Western blot. Yeah, it's different results are possible with antibodies, which I feel like I am a detective every morning when I start going through my mail and I find results from different researchers and I'm troubleshooting it, because it is like a puzzle oftentimes, and in most of the cases it's possible to solve.
Okay, thank you.
You're welcome.
And maybe I can also just briefly add, you spoke a lot about the antibody quality and how to choose the appropriate antibody for the experiment. And I just very briefly wanted to show, how that can be done, at least for the customers in Switzerland, but at least to order. But for the search, of course, all the customers can use this. So since we are a distributor working with many different companies, we can provide a lot of neutral information when looking for the right antibody. So I will just start here on our Agrisera page. That was the link that Joanna provided in the presentation. We have also all the posters and some promotions and some different product information. But I just very briefly wanted to show how that works in our web shop. So let's, for example, when we are looking for Robisco, as an example, we have our application filter. So this works, for all the antibodies. We have hundreds of thousands of antibodies, and what you will find using these filters, you will have the antibodies that were actually validated in Western blot application. And if you look out for these top selling items, these are the international and also the top sellers from Switzerland, that you can very easily also choose an antibody that that it was proven across the globe working well. So this just very briefly. Obviously, you can expand the search always, and if you are not focusing on a supplier, you might have some additional options that you want to look at if you cannot find a suitable antibody from a specific supplier. So yes, just very short. And if you have any questions about the epitopes, for example, and where the antibodies are targeting, feel free to reach out to me, and we can often support you answering these questions.
Yes, and I would like to add that there's, I don't know if you are aware of how many antibodies are available on the market right now. It's around 8 million of antibodies. However, very many antibodies are not validated on endogenous extracts. And this is crucial because the data, be aware that some suppliers are providing data only on recombinant proteins. And there's absolutely no guarantee that if the antibody is recognizing recombinant protein, is going to later recognize an endogenous protein in a tissue. So this is not a validation method. I will talk about it later. But please, when you want to purchase an antibody, carefully study product information sheet. The blot will look beautiful, but if you read carefully the material which was used, it's going to be not a native protein, and you will never be able to achieve such a beautiful fat bands with endogenous proteins for low abundancy proteins as you can for recombinant target. So this can save you a lot of trouble if you keep this in mind for your future purchases. And I would like to thank you today, and I hope you will join our quiz. And I welcome you on the 6th of November for part 2. You can also send me questions by mail until that day. So thank you very much for your participation today.
Thank you very much as well, and see you next time. Have a good day. Bye-bye.